Okay, here we go. Hi, my name is Dr. Anita Bandrowski, and I will be talking to you today about research resource identifiers. And it's primarily about identifying key, mostly biological resources. Thank you. Okay, so looking at what an RRID is, it is essentially a syntax pattern for referencing reagents, things like antibodies. It's a lot like a CAS number, but it is certainly for a lot more of the squishy biological stuff that CAS doesn't really cover. And it's somewhat similar to the IGSN. So the RID syntax, this is the full syntax, which really covers the company or the provider, so in this case, the Developmental Studies Hybridoma Bank, DSHB. There's a local identifier. This is usually a catalog number. The version or the lot of the reagent used, and then the global persistent unique identifier. So this is the RRID component of this, right? If we examine this a little bit more closely, the syntax pattern of this global unique identifier really actually says the following. RRID just says essentially, hey, I used this thing. And the authority or the data source where this information came from is kind of listed in the next component of this. And then that is just a short name of the authority. And then the identifier from that authority is the next portion. So again, somewhat similar to the IGSN. Okay, who are these authorities for biological things? Well, they're people like the MMRRC, and I'll come back to this in a few slides. This is a mouse repository. There are obviously flies, pigs. AdGene has little snippets of DNA. Biosamples, mostly tissue samples. Cellosaurus here has cell lines, and the antibody registry is responsible for antibodies. And so all of these are the authorities, and then their data gets filtered down and gets put into a particular portal called the RRID portal, which makes all of these items fully searchable within a single place. Then authors can be directed to that single place to go and get their RRID numbers. This actually does happen. I'll show you how that happens by the journals. So the journals have instructions to authors that say, hey, go get your RRIDs. Here's where you go. Now, what else is in the RRID portal is a bunch of statements about some of these individual RRIDs. And so the literature itself can reference a particular item, and it can say something like, this is good, this is bad, this is contaminated. And there are a lot of projects like core facilities, international projects that also will say things about some of these different reagents. Okay, so all of that, again, is put into a single portal, which somebody can go in, they can type in their query, and get a list of the matching reagents. Okay, but does it work? Does it work to actually increase at least the ability for somebody else to come in and find those reagents? Well, we did a 3-month pilot project. We're still going. This is now our 7th year. With 25 journals, and we found out that essentially without RRIDs, authors are fairly poor at actually identifying the resources that they used, but with RRIDs, they're much, much better. So in this bar graph, we're just looking at the identifiability of these reagents, and so for antibodies here, in the issue of the journals, right before we started asking authors to provide their RRIDs, about half the time you could figure out which antibody the authors used. But as soon as we started asking for these RRIDs, remarkably enough, that goes way up to about 90%. And these antibodies, they're a very tricky reagent, and the companies that are involved in making and selling and producing these antibodies or organisms or tools are also very interested in making sure that they know very well, how their stuff is being used, so they can maybe yank it off the shelf if needed, or they can add additional validation data. And so here's one of those companies. It's one you may have heard of, Thermo Fisher Scientific. They're a fairly big company. But what they did is they said, hey, look at our lovely list of 115,000 antibodies, and we're going to make sure that anybody who wants to find a citation can actually find that citation right on our website. And of course, so they really encourage the RRID by using it on their website. So we looked across time, and now this is a study of about 2 million papers. For these antibodies, for these antibody-containing papers, there were about 360,000 of these. And we looked all the way back to 1997 and just found the percentage, essentially, of these antibody citations that were associated with a catalog number, which makes them findable again. And it was about 10 to 15% back in the 90s. And you can see that this blue line, this is the overall literature, actually increases to about 25% somewhere in the 2015-2016 time period. And then there's really this nice big deflection towards more identifiability, towards more catalog numbers. And when we looked a little bit deeper at the individual journals, so now you've got a bunch of these individual lines, you can see that there are a few journals, like here, Cell and eLife, that are really driving this inflection. And those journals, what they've done in 2016, both of them came on in 2016, you can see that when they changed their policies and they started to enforce RRIDs, they changed from kind of an average journal to 90+% identifiability. The PLOS One instructions to authors also changed, but nobody reads those. So, but certainly there is even a little bump there in 2018, 2019, even in PLOS. Okay, whoops. All right, so what does this actually look like in a paper? So if I were to run across a paper and it had RRIDs, how would it look? So here is a method section, a partial method section from this eLife paper. And again, what we have is a bunch of mice. These are all mice, and I know that because of the prefix of the identifier. So this is Jackson Laboratory, and that's the MMRC. So if I click on one of those, through the magic of the internet, I end up magically through the RRID resolver on the mouse strain datasheet, which, by the way, also encourages the use of RRIDs. So it all kind of comes full circle, doesn't it? Okay, but why would I care about this thing? If you know that there are lots and lots of product recalls, even lettuce, which, as far as I can tell, is not barcoded, can be recalled. In fact, it was fairly recently. But does this really happen? Can papers be recalled because of a reagent? Well, not really, but maybe they should be. So this is a relatively recent paper. This is actually a story about a paper that found a couple of really nasty bugs in some Python scripts that were used in hundreds and hundreds of chemistry papers. And essentially it affected NMR shifts. And the problem was that there were incorrect measurements that were being made, and those depended on the operating system. So this is a very serious problem, and there may be other problems. So again, these Python scripts are listed. They have an RRID, and they have a lovely warning that says, hey, by the way, there's a paper. You might want to check this out. There's potentially a problem. Good things are sometimes said about these reagents. So this is that information about each RRID. This is a note that the ENCODE project actually validated this particular antibody. And here's another note, this one actually in red now, that says, hey, this is a contaminated cell line. But the question is, do people read these notes? Do they? Yep, they do. So here we found 150,000 papers that did not use RRIDs, and then we ran a fairly strict matching algorithm to try and figure out if these cell lines were actually matched one of the cell lines that were listed in the problematic list. And we did that, and we found about 16% of the papers actually were using one of these problematic cell lines. Now, again, some of these are fine to use, but many are not. So when we looked at the 634 papers that were published with cell line RRIDs at the time, it's way more now, but these papers only found, matched 5% of the time. So with no additional editorial oversight, without somebody, beating people's heads in with don't use these contaminated reagents, simply the note there. And by the way, the note wasn't red. This note wasn't red. We recently turned it red to make it more obvious. But during that study, the note wasn't red. It was just all black text. And it seemed like there was a 66% reduction of the use of these naughty cell lines without additional oversight. So really, this is saying that when people have to go into a particular portal, they have to go look up a number, they are paying attention. They are looking at what is stated about those reagents. Okay, but this whole process still is very difficult, right? We still don't have more than about 30,000 papers using RRIDs at the moment, and we have been at this for over 6 years. So can we actually automate addition of RRIDs into manuscripts before they're ever published? And so the answer to that came from an automated tool that we've been working on for the last couple of years called Sciscore. This is an automated reviewer. It takes as input the methods section of a paper, and it gives as output a set of helpful hints, hopefully helpful hints in the report. It is currently in use in about 12 different journals, where the authors put in the journal submission system, they put in their method section, and they get back a report. And so what this thing does is it actually provides authors with relevant RRIDs if the tool can find them, or notes where the RRID should be if the tool cannot suggest an RRID. Cell line problems, if those are found, it tries to add those right to the report, and it does also turn those red. It turns broken GitHub links and other DOIs that are broken red. It tries to point out clinical trials issues where different database identifiers are actually not proper. So it does all those things, and it helps authors take out some of these problems in the literature. Okay, so AdGene came to us a while back, and AdGene is a big supporter of RRIDs with each of these pages of their phages. They have a lovely how to cite this plasmid section, which of course includes the RRID. But they said, hey, we spend a huge amount of time actually soliciting plasmid deposits. They have a ton of them, but there are lots more coming out. These things are created by a lot of different labs, and they have to be sent to AdGene to store them, and then also to redistribute them. So one of the questions that we got is, hey, can we detect plasmids in the text? And then can we encourage authors to actually deposit those or any of the other reagents that might be used? How can we potentially work either with the journals or work with some tools like Sciscore in order to be able to encourage these good behaviors. And I think that the question to that, or the answer to that is, of course, but we don't really know how to best do that at this point. So we're trying to form this working group in order to try and figure out how is it best addressed in the scientific literature process. Okay, so thank you very much for listening. And I would always encourage everyone to use RRIDs in everyone's next paper if it is appropriate to do so, or using the IGSNs if you happen to really like rocks. Okay, if you use the IGSNs, we probably will not tweet about it. But if you use an RRID, we actually do have a robot Twitter account, which will tweet about your paper, which is kind of just a fun thing that I am now talking about. Okay, thank you very much and have a good day.